Good evening. Kindly give me message and confirm that I am audible to all. The students and viewers who are connected, kindly give me message and confirm that I am audible to all. That means my sound is reached to you. मेरी आवाज अगर पहुंच रही है तो थोड़ा कंफर्म कर दें. Yes, I am audible. And now I am initiating the topic which I am proposed for today, and the topic which is very simple, and probably you all have well read and familiar to the topic, that is the glycolysis, and the another part is the anaerobic 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 respiration, that is pentose phosphate shunt, or otherwise it is referred as the glucose phosphate shunt, or it is also referred as the pentose phosphate shunt. And as I think that you well known that the both glycolysis and glucose phosphate shunt are completed in the absence of oxygen. That is an aerobic mode of the energy production, and they are related with the anaerobic mode, anaerobically production of the energy in the biological system. Of course, it is necessary to say that the efficiency of the anaerobic productivity of energy is energetically less efficient than the oxidative mode of the energy production. And it is the theoretical part, but I am describing here the different steps, and that steps are necessary for you to remember the steps. And for the remembering of the steps, it is necessary to keep the steps of the glycolysis and phosphate shunt, pentose phosphate shunt in your memory. So I am just at first going to explain the glycolysis, or it is also referred as Embden Myer half pathway, on the basis of the name of workers responsible for the deciphering of the different steps. Initially, in the glycolysis, 1 mole of glucose is used, and by the use of 1 mole of glucose, the enzyme 11 enzyme complexes. It is necessary to remember that 11 uh, enzymes complexes are associated, and it is responsible for the production of 2 molecules of the lactic acid. And in another way, in the yeast, it may be in yeast, it is responsible for the production of 2 units of ethanol plus 2 CO2, 2 units of carbon dioxide. So, if we observe the glycolysis, the all steps, 11 enzymatic steps of the glycolysis, we divided the complete steps of the glycolysis into two parts, broadly into two parts. That is, the first part is endergonic process. The first part is endergonic process, and it consumes 2 molecules of ATP. And in the endergonic part, glucose is converted into fructose 1,6 diphosphate. So the steps involved in the endergonic process of the glycolysis is glucose, and glucose is converted into glucose 6 phosphate. Now the glucose 6 phosphate is converted into fructose 6 phosphate, and fructose 6 phosphate is converted into fructose 1,6 diphosphate. So it is very easy to remember the steps. That is, the glucose converts into the glucose 6 phosphate. Glucose 6 phosphate is converted into the fructose 6 phosphate, and fructose 6 phosphate is converted into the fructose 1,6 diphosphate. And here, the two at the 2 points we observed the phosphorylation. One, that is glucose is phosphorylated to form the glucose 6 phosphate, and glucose 6 phosphate is converted into the fructose, that is the isomeric form. Fructose is the isomeric form of the glucose, and again the isomeric form of glucose, that is the fructose 6 phosphate, is again phosphorylated, and it converts into the fructose 1,6 diphosphate. If you remember the steps and if you keep in the mind, then you analyze the enzyme. And when the molecule is phosphorylated, it uses the kinase enzyme. And when it is isomerylated, it is responsible for the use of isomerase enzyme. So now it is very simple that you remember that glucose is converted into the glucose 6 phosphate in the presence of glucokinase or hexokinase enzyme. Now the glucose 6 phosphate is converted into the fructose 6 phosphate by the glucose 6 phospho isomerase enzyme. It is very simple. You supplement the name of the enzyme, chemical action with the substrate. The third one is the fructose 6 phosphate is converted into the fructose 1,6 diphosphate. So one step again phosphorylated. So here the enzyme involved is named as or is referred as fructokinase. In this way, one molecule of glucose is converted into the one molecule of fructose 1,6 diphosphate after utilization of or after consumption of 2 molecules of ATP. So the 2 molecules of ATP is converted into the ADP. And this one is the endergonic process or endergonic pathway of the glycolysis. Here, particularly in man or particularly in the animals, the glucose is stored in the form of glycogen, particularly in the muscle and liver. So, the a stored component which is responsible for which is related with the production of energy is glycogen. And one thing important which is necessary to always remember, because sometimes students did wrong, because glycogen is not converted into the glucose during the anaerobic pathway, but glycogen is converted into the glucose 1 phosphate. And here the glucose 1 phosphate is formed from the glycogen by the use of glycokinase enzyme in the presence of ATP, and that ATP is converted into the ADP. And now the glucose 1 phosphate is converted into the glucose 6 phosphate. That is the rearrangement, internal rearrangement of the phosphate group. That is the rearrangement. Here it is the mechanism is the rearrangement. So the glycogen is not entered by the glucose in the glycolysis, but glycogen is entered in the glycolysis, in the pathway of glycolysis through glucose 6 phosphate. And it is my suggestion and request also to understand properly the glycolysis, because if you are not understanding properly the mechanism of glycolysis, then you feel problem in the proper understanding of the pentose phosphate shunt, which I am going to explain just after the completion of the glycolysis. So it is the part, it is the first part. Now the fructose 1,6-diphosphate of the endergonic pathway is entered in the exergonic pathway, and the first step which is responsible for the conversion of the fructose 1,6-phosphate, that is the 6 carbon molecules, into 2,3 carbon molecules, that is dihydroxyacetone phosphate and glyceraldehyde phosphate. And the mechanism and the process is referred as aldol condensation. And as I think probably you studied aldol condensation in the mechanism of organic chemistry. So here the 6 carbon fructose 1,6-diphosphate is condensed, that is the cleaved or condensed. Why so it is referred as the aldol condensation, into 2 molecules of the 3 carbon compounds, that is dihydroxyacetone phosphate and glyceraldehyde phosphate. Now the dihydroxyacetone phosphate is again converted into the glyceraldehyde phosphate. So the 1 molecule of glucose is responsible for the synthesis of 2 molecules of glyceraldehyde phosphate after utilization of 2 molecules of ATP. Now the pathway which carries forward is the exergonic pathway. And I am describing the one unit of the glyceraldehyde phosphate, but because here the one glucose is responsible for the production of two molecules of glyceraldehyde phosphate, so both represent the same pathway. And the process which is associated with the exergonic part is oxidoreduction process. Just in my last class, I explained you the electron potential or oxidoreduction potential, and I said that the changes in the oxidoreduction potential or electron potential is responsible for the release of energy, and that free energy released in the system is responsible for the formation of high energy phosphate group. So here, because here the oxygen is absent and it is not carried to the electron transport system and it is not carried to the Krebs cycle, so at the substrate level or by the anaerobic process, the energy is also produced. Of course, the efficiency of the production of energy and is less than the oxidative pathway, because the less energy is released in the aerobic condition. And here, the one unit of glyceraldehyde phosphate is responsible for the synthesis of 3-phosphoglycerate through 1,3-diphosphoglyceraldehyde or glycerate. I am again repeating that the glyceraldehyde phosphate produces 3-phosphoglycerate through 1,3-diphosphoglycerate. Why I am representing in this complex manner? Here some fact is present. So I am representing here in some complex manner. If I want to say in simple way that I say that glyceraldehyde phosphate is converted into 1,3-diphosphoglycerate in the presence of NAD associated dehydrogenase enzyme, and thereafter the 1,3-diphosphoglycerate is converted into 3-phosphoglycerate in the presence of enzyme phosphoglycerol kinase and synthesize the one unit of ATP at substrate level. But no, I am not asking like this. I am saying in some complex way. And what is complexity? I am saying that the glyceraldehyde phosphate is synthesized 3-phosphoglycerate via 1,3-diphosphoglycerate in the NAD associated dehydrogenase enzyme complex and produces one unit of NADH 2 or NADH plus H plus and one unit of ATP. Because why I am saying like this? Because the energetics, if we observe the energetics, because here the one ATP is synthesized, and if the one ATP is synthesized, that the energy, some energy must be released in the system in free state, that is the free energy of the system, which is responsible for the synthesis of one high energy phosphate group, pyrophosphate group, PI, that is represented by PI, pyrophosphate group. And here, if we observe this way, the energetic value of the glyceraldehyde phosphate is 12 kilocalorie. Sorry, 12 calorie. And now the energetics of the 3-phosphoglycerate is 4 kilocalorie. Then the 12 kilocalorie is converted into the 12 kilocalorie molecule, biochemical molecule is converted into the 4 kilocalorie biochemical molecule. And here the energy is released. That energy is utilized by the pyrophosphate group to develop the high energy bond, and the energy is utilized in the synthesis of the ATP from ADP. And in this way, the enzyme complex is associated, that is the dehydrogenase, because the NAD, that is the H plus, is released, that is proteinated. It is the proteinated. That is one, when it is proteinated, then the proteination of the system is neutralized by the release of energy, and that release of electron, and that electron moves from the high energetic potential to the low energetic potential. And in this way, some free energy is available in the system. And then the dehydrogenase is responsible for the synthesis of NADH plus H plus. And that proteination is responsible for the movement of the electron from the higher energetic potential to the lower energetic potential. So here, the energy is developed by the movement of proteination and movement of electron. Now, the 3-phosphoglycerate is converted into 2-phosphoglycerate. And here, the enzyme is referred as mutage, because it is the rearrangement. The phosphate, pyrophosphate of the third position of carbon is rearranged to the second position of the carbon. So the 3-phosphoglycerate is converted into the 2-phosphoglycerate. Now the inhalation is occurred, and 2-phosphoglycerate is converted into phosphoenol pyruvate. And again, when the phosphoenol pyruvate or 2-phosphoglycerate is converted into the pyruvate through phosphoenol pyruvate, here 1 unit of ATP is also produced. But here we observe on the upper, in the just before, when the glyceraldehyde phosphate is responsible for the production of 3-phosphoglycerate, here the proteination is observed, that is H plus is released due to the presence of NAD linked dehydrogenase, and that H plus, release of the H plus, is neutralized by the movement of electron from higher energetic level to the lower energetic level, and that the free energy is trapped by the system, and it converts into the high energy pyrophosphate bond. But here, when the 2-phosphoenol glycerate is synthesized pyruvate through phosphoenol pyruvate in the presence of enolase and kinase enzyme, the electron rearrangement is occurred. And that rearrangement during the inhalation, particularly during the inhalation, the rearrangement of electron is observed, and that electron rearrangement is occurred to conserve sufficient potential energy, and that potential energy is converted in the form of the ATP. Now, the NADH2 formed at the stage of the glyceraldehyde 3-phosphate to 3-phosphoglycerate is utilized by the pyruvate, and now the pyruvate is converted into the lactic acid by the by the utilization of NADH2, and now the NADH2 is converted into the NAD, that is the reduced form of the nicotinamide adenosine dinucleotide is converted into the oxidized form. So here the second stage is the oxidoreduction process. The second stage of the glycolysis is the oxidoreduction. Now the oxidoreduction process is by the oxidoreduction process, it is a energy conserving mechanism. It represents a energy conserving mechanism, and in the one glyceraldehyde phosphate step, it produces the 2 ATP. So the one molecule of glucose is responsible for the production of 4 molecules of ATP. And in the oxidative condition, the pyruvic acid, it stops the further reaction from pyruvic acid, and pyruvic acid moves to the moves to form the coenzyme A to form the acetyl coenzyme A. So if it is in the oxidative condition, in that condition 4 molecules of ATP and 2 molecules of NADH are formed. That 2 molecules of NADH are responsible for the carrying of received protein or hydrogen or H plus to the electron transport system. And if the oxygen is not present, then it is responsible for the production of 2 units of ATP and 2 units of lactic acid. So in this way, the glycolysis is responsible for the production of lactic acid, or in the condition of the alcoholic fermentation, the lactic acid moves one step further and it is responsible for the production of 2 units of ethanol and releases the 2 molecules of CO2. So we studied here the mechanisms of glycolysis. Now the next part, and as I think that you all who are associated presently in my life have properly understand the mechanisms of glycolysis, and if if anyone has any problem, then you give me message, because the glycolytic steps is associated with the pentose phosphate shunt or glucose phosphate shunt और हेक्सोस फॉस्फेट शंट, जो भी आप नाम ले सकते हैं, बोल सकते हैं. तो इफ यू हैव एनी प्रॉब्लम इन दीज स्टेप्स विच आई एम जस्ट डिस्क्राइबिंग, तो देन यू गिव मी मैसेज, 1 or 2 मैसेज, or otherwise you give me message that all are okay, and now I move one step further, because I want to give you informations for the hexose phosphate shunt or glucose phosphate shunt also. Someone mentioning that no problem. Okay. Someone mentioned that please describe it in short. Okay. Just I am describing. Suppose you only want to remember the steps. 11 steps. Just I am describing in brief. The glucose. Glucose is converted into the glucose 6 phosphate. Glucose 6 phosphate is converted into the fructose 6 phosphate. Fructose 6 phosphate is converted into the fructose 1,6 diphosphate. Fructose 1,6 diphosphate is represented aldol condensation and converted into the 3 carbon 3 glyceraldehyde phosphate. The 3 glyceraldehyde phosphate is converted into the 1,3-diphosphoglycerate. 1,3-diphosphoglycerate is converted into the 3-phosphoglycerate. 3-phosphoglycerate is converted into the 2-phosphoglycerate. 2-phosphoglycerate is converted into the phosphoenol pyruvate. Phosphoenol pyruvate is converted into the pyruvate, and pyruvate is converted into the lactic acid. In this way, the glucose from glucose to 3-carbon glyceraldehyde phosphate is the exergonic process. Endergonic process, and it utilizes 2 molecules of ATP and the enzymes associated with the kinase where the ATP is utilized, and isomerase when the glucose is converted into the fructose 6 diphosphate. Here, the next step is the exergonic pathway, where the energy is released. The exergonic pathway is oxidoreduction pathway, and here the 3-phosphoglycerate pyruvate to the synthesis of the lactic acid is included in the exergonic pathway, and here 2 molecules of NADH2 and 4 molecules of ATP are synthesized from 1 molecule of glucose. But if we calculate the 1 glyceraldehyde, then 2 units of ATP and 2 units of NADH plus synthesized when when oxygen is present, and pyruvic acid is converted into the acetyl coenzyme A. But where the oxygen is not present and pyruvic acid is converted into the lactic acid, here from the pyruvic acid to lactic acid, the NADH2 is absorbed, and that means the 1 glucose produces 2 units of the lactic acid and 4 molecules of ATP. So, just I am describing the brief, and when we move in the pentose phosphate shunt or glucose phosphate shunt or hexose phosphate shunt, whatsoever you free to say, then it is also the anaerobic process and alternative pathway of the glycolysis, which we are going to just describing. And in this condition, in the glucose phosphate, because the glycolysis is also occurred in the cytoplasm. Of course, Krebs cycle and electron transport system is occurred in the mitochondria, but glycolysis is occurred in the cytoplasm, and Embden Myer popularly known as Embden Myer pathway. Now, in the hexose phosphate shunt, the main site, which is where the glucose phosphate shunt or hexose phosphate shunt or phosphate shunt is observed is the RBC. So it is associated with the microevolution and protection with respect to the some evolutionary except phenomena is associated, which we will discuss during the microevolution, and it is associated with the development of the anaerobic respiration in the RBC, because in the RBC of man, the membranous cell structures are secondarily lost. So nucleus is not present and any membranous is not observed in the mammalian RBC. So in the RBC, the energy is produced anaerobically because the mitochondria is not present, anaerobically by the glucose phosphate shunt or hexose phosphate shunt pathway. And the pathway, the glucose phosphate shunt pathway was elucidated by Bernard Haerker, the worker which is responsible for the elucidation of the hexose pathway is Bernard Haerker, and in majority of organisms, the glucose phosphate shunt is observed in the cytosol. Of course, it is observed in the mammalian RBC, and in plant, it is observed in the plastids. Here, the glucose phosphate shunt is also divided into two parts. One is the oxidative and another is the non-oxidative. Likely we observed in the glucose pathway also, glycolysis pathway also. The first part is responsible for the phosphorylation, and that the enzyme is involved is the kinase. But the second part, after the aldol condensation, we studied just at present that the NADH is associated, and that is responsible for the oxido reduction. And when the energy exchange is occurred, the energy exchange in the biological system is always requires oxido reduction pathway or oxido reduction process. In last class, I described in details that what is the matter, why electron moves, like all this. And here, the oxido reduction is occurred, but the oxido reduction is occurred in the absence of oxygen. That means the oxido reduction is related with the movement of H plus, that is proteination. Then in the glycolysis also, the proteination is responsible for the oxido reduction. And when any element is proteinated, that proteination is compensated by the movement of the electron. And when the electron moves from the higher energetic level to the lower energetic level, it is responsible for the change of energy, either in the plus or in the minus side, which we all discussed in my various last classes. So here, the glucose 6-phosphate, glucose, that is a two-part in the glucose phosphate shunt or hexose phosphate shunt or pentose phosphate shunt. We use the term pentose phosphate shunt because the pentose sugars are also related with the in this process, in the shunt process or in this process. So the glucose 6-phosphate I am at first describing the oxidative part of the glucose phosphate shunt, and in the oxidative part of glucose oxidative part of the pentose phosphate shunt, 1 unit of glucose 6 phosphate is converted into 1 unit of ribulose 5 phosphate. So ribulose is the 5 carbon compound, and here in ribulose 5 phosphate is initiated the non-oxidative pathway of the pentose phosphate shunt. So the ribose 5 phosphate is associated with the non-oxidative steps. Here the glucose 6 phosphate is converted into 6-phosphogluconolate. Sorry, 6-phosphogluconolactone. Lactone component is present, and here the enzyme is associated with the NAD-linked dehydrogenase. That means NADP linked dehydrogenase enzyme is responsible for the proteination, and NAD oxidized form of the NAD is reduced as the NADPH2, and that the removal of the hydrogen from the glucose 6 phosphate is responsible for the synthesis of 6-phosphogluconolate glucone 6-phosphogluconolactone, and 6-phosphogluconolactone is again hydrogenated, मतलब contains receives OH molecules from the H2O in the presence of enzyme glucolactonase, because it is the name of substrate, that is 6-phosphogluconolactone, and from the glucolactone, the name of enzyme is gluconolactonase, and then gluconolactonase is responsible for the synthesis of 6-phosphogluconate, and then 6-phosphogluconate is again dehydrogenated in the presence of NADP associated dehydrogenase enzyme, and now the NADP is protonated to form NADPH2. So the two molecules of NAD is reduced, one at the site of glucose 6 phosphate to 6-phosphogluconolactone, and one at the 6-phosphogluconate to ribulose 5 phosphate. And here the 6-phosphogluconate, after protonation in the presence of NADP linked dehydrogenase, synthesize ribulose 5 phosphate. Because here the NADP is associated, that is the oxidized form, and that is the couple. In last class I already represented that they are the coupled structure, redox coupled structure, NAD P is converted into the NADPH2. That is the oxidized form is converted into the reduced form. Likely here the two molecules in the oxidative process, the glucose 6-phosphate produces 1 unit of ribulose 5-phosphate and 2 units of NADPH2. Now, from onward, you remember and keep the facts in your mind. It is necessary because the ribulose 5-phosphate is the pentose sugar, phosphated form of the pentose sugar, and here the two enzymes are involved. One is the isomerase and another is the xylulose 3-epimerase, 3-epimerase. And I already described what is the epimer during the isomerization in the carbohydrate. So here, the one mechanism is the isomerization and one mechanism is the epimerization. So the ribulose 5-phosphate synthesizes ribose 5-phosphate. Ribulose is the ketonic form and ribose is the aldehydic form. That means ribulose 5-phosphate and ribose 5-phosphate is the isomeric form. So ribulose 5-phosphate, that is the ketonic state of the 5-carbon sugar, is converted into the ribose 5-phosphate in the presence of enzyme isomerase. In another way, in another hand, the ribulose 5-phosphate forms epimer xylulose 5-phosphate. It is the epimer. So the ribulose 5-phosphate is converted into the xylulose 5-phosphate in the presence of enzyme epimerase. So the ribulose 5-phosphate synthesizes 1 unit of ribulose 5-phosphate and ribulose 5-phosphate synthesizes xylulose 5-phosphate. Now, the xylulose 5-phosphate and ribose 5-phosphate both are condensed. Both are condensed because the ribulose 5-phosphate has 5 carbon, xylulose 5-phosphate has the 5 carbon. That is the total of the 10 carbons. Have you any problem? Probably, I think that you have no any problem because ribose 5-phosphate has the 5 carbon and xylulose 5-phosphate has the 5 carbon. And 1 unit of the ribulose 5-phosphate and 1 unit of the xylulose 5-phosphate are condensed, reorganized in the presence of enzyme transketolase, because both are the keto group, transketolase, and form 1 unit of 3-carbon glyceraldehyde 3-phosphate and 1 unit of 7-carbon pseudohaptulose 7-phosphate. That is a 10, 7 and 3. That means 1 unit of ribulose 5-phosphate and 1 unit of xylulose 5-phosphate condensed in the presence of enzyme transketolase, and 1 unit of 3-carbon glyceraldehyde 3-phosphate is formed and 1 unit of 7-carbon they do have to load 7 phosphate is synthesized or formed. Here, the 1 unit of glyceraldehyde 3 phosphate, which is synthesized here, moves to the glycolysis. Moves to the glycolysis. And now the steps remaining after for the synthesis of lactic acid from the glyceraldehyde 3 phosphate is completed. Now the they do have to load 7 carbon, they do have to load phosphate is again reorganized and form fructose 6 phosphate and erythrose 4 phosphate. Fructose 6 phosphate and erythrose 4 phosphate. Again, fructose 6 phosphate can move to the glycolysis. Again, one component is here, that is erythrose 4 phosphate, and erythrose 4 phosphate combines with the xylulose 5 phosphate, that is 6 carbon xylulose 5 component, and here the enzyme transketolase and transketolase, the enzyme transketolase, the presence of the enzyme transketolase converts 1 unit of erythrose 4 phosphate plus 1 unit of xylulose 5 phosphate into glyceraldehyde 3 phosphate and fructose 6 phosphate. Again, here the glyceraldehyde 3 phosphate and fructose 6 phosphate move to the glycolysis and complete the anaerobic respiration. So, the glucose phosphate shunt or phosphate pentose pentose phosphate shunt are supplemented. It is not the new or anything else, but they supplemented the process of glycolysis or anaerobic respiration. So the Embden Myer pathway and pentose phosphate shunt both are included in anaerobic energy production of cell. So I suppose that something in some explanations are of course new here, but I think that you all understand properly the lecture delivered today. But if any feels any problem, then you ask any question, or if you any problem, you give me message. I am just observing your message, and if you feel any problem, give me message. If you have no any problem, then give 1 or 2 messages that okay. No one can mention. Okay. Now we receive the message that no problem. So thank you to all. Thank you for watching of my lecture. Now I will again communicate the topic I will discuss on tomorrow or day after tomorrow. So thank you to all.